So next we're going to switch gears and we're going to talk a bit about the immune system. And first we're going to talk about the non-specific, or what's called the innate immune response. Innate means built in, you're born with it. So this protects you against general pathogens. These are mostly barriers. So we're just going to talk about 2 or 3 important parts of the non-specific immune response. The most important part is the skin. It acts, first of all, as a physical barrier, but it also secretes chemicals like urea and sweat, which help to kill pathogens. Your stomach acid, mostly it's hydrochloric acid. The pH in your stomach is usually somewhere around 2. So most things that enter your stomach are made of mostly water and protein. When the proteins hit that stomach acid, they denature, and that means they're no longer inactive. So stomach acid inactivates or kills most things that enter into your stomach. We're going to skip down and talk about inflammation next. So when you're burned or cut or scratched, cells in that damaged area, called mast cells, release these chemical signals called histamines. Think of these like SOS signals. And one of the types of cells that they affect the most are the cells in the blood cells nearby. They cause the blood vessels nearby to dilate. That sends more blood to the damaged area, which means that area gets more platelets to help cause a clot. And it also gets more white blood cells to help to deal with invaders that might have penetrated through the torn area. The normal flora. Even though the word flora usually refers to plants, when we're talking about the body, it refers to the bacteria that normally live in your body. These bacteria help to block off other things from growing. So think of them sort of like your offensive line. A lot of people get kind of secondary infections after they take antibiotics because the antibiotics kill off a lot of these normal flora, and they allow things like yeast, which normally couldn't grow very much because of all the bacteria, to grow. We'll also mention saliva and tears. Both contain enzymes called lysozymes, which help to break down bacterial cell walls, and that's why you very rarely will get infections in your mouth or in your eyes. So these are all non-specific parts of your immune response. But the more important thing we need to talk about is the specific immune response, which we'll also refer to as the acquired or adaptive immune response. So this allows you to build up an immunological memory after an infection, and it essentially allows you to become immune to that same antigen when you're exposed to it multiple times. So 2 keywords. An antigen is a foreign substance, usually a protein, which can stimulate your immune response to initiate an immune response, or your immune system rather, to initiate an immune response. An antibody, on the other hand, is a protein made by your body, made by the plasma cells, a type of B cell, in response to an antigen. They bind to very specific antigens, and in one way or another, they help your body to neutralize and get rid of that antigen, that invader. So there's 2 parts of the specific immune response we'll talk about. One of them is called antibody mediated, or sometimes humoral immunity, and the other is cell mediated. The antibody mediated immunity helps you to deal with germs that are in your body's fluids, in the blood and the interstitial fluids and the lymphatic fluids. That's why it's called humoral. Humoral refers to body fluids. This is true because antibodies are these big, bulky proteins, and if a pathogen is already inside a cell, the antibodies can't get in there because they're proteins and they're large and they're charged. On the other hand, the cell-mediated immune response involves a type of cell called a cytotoxic T cell, which helps to destroy your own cells that are infected with internal pathogens, intracellular pathogens. And this helps to it helps to lower the spreading of the germ within your body. So let's talk a bit about antibody-mediated immunity. So typically the way this gets started is a macrophage, a white blood cell, will phagocytize an invader, a pathogen. And it will use the lysosomes to intracellularly digest that pathogen. It will take some of the proteins, some of the antigens from that pathogen, and it will display them on its own cell membrane, on a type of protein called an MHC II complex. This process of presenting these antigens is called antigen presentation. And it presents them to a type of cell called a helper T cell, or T helper cell. So only T helper cells that can bind to that specific antigen will connect. They physically connect with the macrophage. And we can see that happening. We'll find a picture here in a minute, hopefully. Maybe I don't have that picture. So they bind together, and that activates the helper T cell. That helper T cell will then start to release cell signals, particularly a type of chemical called interleukins. And those activate B cells, another type of white blood cell, specific to that antigen. And the first thing it tells those specific B cells to do is to clone themselves, make more copies of yourself. Most of those B cells transform into what are called plasma cells, and they become factories for making antibodies. A few of those plasma cells, on the other hand, become activated memory cells. They don't make antibodies, but they stay in your body typically for life. Once the antibodies get made, they attach to the antigens. They cause the body to help to alleviate that infection and get rid of it. And then after a certain amount of time, all those plasma cells are programmed to essentially destroy themselves. They go through a process of programmed cell death called apoptosis. But the memory cells don't go through that process. They stay around. And if you ever experience that same exact infection again, they will allow you to skip some of these first few steps, and they'll allow your immune response to be so much bigger and so much faster that essentially you don't have any symptoms the second, the third, the fourth time you're exposed to that same thing. And this is why we say you're immune after having fought through one infection. So here we can see those steps. So the macrophage engulfs the germ, it breaks it down, it displays its antigens to a helper T cell. The helper T cell releases interleukins, let's go this way, which activate B cells specific to this antigen. They clone themselves. Most of them transform into plasma cells and make antibodies, which attach to this antigen and help your body to get rid of it. Some of them remain as memory cells to help you become immune to this thing in subsequent exposures. Notice that's also true of the memory helper T cells. While we're here, let's go ahead and talk about, and also notice that the same antibody-mediated immune response can be activated by antigens just floating around in the body fluids that happen to attach to B cells specific to those antigens. So some of these steps don't have to happen here in the middle. All right, let's talk about cell-mediated immunity while we're here. So again, the purpose of cell-mediated immunity is to kill your own cells that have an internal infection. So this can start off the same way as antibody-mediated immunity. A macrophage engulfs a pathogen, presents antigens to helper T cells. Those helper T cells release cell signals that stimulate cytotoxic T cells. An important thing to know is that all your cells have this complex called an MHC1 complex that they display proteins from inside themselves on. If they have an internal infection, instead of displaying normal proteins, they display maybe viral proteins or bacterial proteins. The cytotoxic T cell can pick up on those, and essentially that's a signal to the cytotoxic T cell that it needs to destroy that cell that is presenting bacterial or viral antigens. And it will do that essentially by puncturing it, by perforating it, poking holes in it, killing the cell. So once the helper T cell sends out these signals, just like what happened over here, it causes the cytotoxic T cells specific to this antigen to clone. Most of those cells become active cytotoxic T cells, and they go out looking for your cells that are infected and killing them. After a certain amount of time, once the infection's over, they go through apoptosis and they essentially die. But once again, there are memory cells left behind. All of these memory cells help to make your immune response to that same germ the next time so much bigger, so much faster that you don't get sick. Let's go ahead and talk about one other topic here related to antibodies. Switch gears. We only have a few genes that code for antibodies, but we may need to make up to 10 to the 12th different kinds of antibodies in our life. So how can only a handful of genes code for this many different distinct antibodies. It has to do with the idea of alternative DNA splicing. So here we have a gene for an antibody. Remember that the coding parts of the gene are called exons, and that in between those coding parts are these segments that we used to think of as junk DNA called introns. But what we found is that these introns are important in regulating the genes and allowing one gene to actually act like multiple genes. So the DNA gets transcribed into RNA. Both the exons and the introns are transcribed. But in eukaryotes, after transcription, we have this step called RNA processing or alternative splicing. And what happens typically is that these intron sequences are removed and the exon sequences are spliced back together to make an active mRNA, which then gets translated into a protein, in this case, an antibody. But notice that sometimes the introns are removed, but sometimes certain exons are also removed. So in this case, notice the purple exon, exon 3, got removed. So that even though these RNAs both came from the same gene, they're different. And they code for different proteins, in this case, different antibodies. Notice here's a third case where the same gene codes for a different protein or antibody. So one gene can actually code for multiple different proteins, or in this case, multiple different antibodies. And that's why we can respond to so many different kinds of infections. So let's also talk about the primary versus the secondary immune response. The primary immune response happens the first time you're exposed to a particular antigen. The response is kind of slow. We've got all those complicated steps that we talked about earlier that have to happen. And the number of antibodies made is pretty low. So notice it takes several days to make the antibodies. The amount of antibodies never gets all that high. By the time the antibody level gets to about right here, you're probably over the infection. So most of those plasma cells start to die and your antibodies levels drop down to essentially zero. All right, so let's say years later, you're exposed a second time to the same exact antigen. This time you have all those memory cells that were created during the primary immune response. And they allow you to skip a bunch of those initial steps. They make the immune response so much bigger and so much faster that you don't get symptoms the second time. We call this immune response the secondary immune response. And notice how many more antibodies are made and how much steeper this line is. That tells you that the rate of making those antibodies is so much faster. All right, so that's it for the immune response and the intro to cell signaling.